Elaboration of the Central Dogma by Jordan, Neve, and Andrew. In 1869, Swiss chemist Friedrich Miescher identified what he called nuclein inside the nuclei of human white blood cells. Nuclein later changed to nucleic acid and eventually to deoxyribonucleic acid, or DNA. Miescher's discovery wasn't valued for 50 years. Russian biochemist Phoebus Levine discovered the order of the major components of a nucleotide, being phosphate, sugar, base. He discovered the carbohydrate component of RNA, which is ribose, and DNA, which is deoxyribose. He correctly identified how RNA and DNA molecules are put together. Austrian biochemist Erwin Chargaff expanded on Levine's work. First, he found that the nucleotide composition of DNA varies among species, and second, he concluded that almost all DNA maintains certain properties, even as its composition varies. Chargaff's rules state that the total amount of purines, adenine and guanine, and the total amount of pyrimidines, cytosine and thymine, are usually equal. Chargaff's realization that adenine binds to thymine and cytosine binds to guanine, combined with X-ray crystallography work, contributed to James Watson and Francis Crick's derivation of the three-dimensional double helical model for the structure of DNA. Since 1953, their model has four major features. 1. DNA is a double helix connected by hydrogen bonds, and adenine base pairs with thymine and cytosine base pairs with guanine. Second, most DNA double helices are right-handed. Third, the double helix is anti-parallel, which means that the 5' end of one strand is paired with the 3' end of its complementary strand, and vice versa. Nucleotides are linked to each other by their phosphate groups, which binds the 3' end of one sugar to the 5' end of the next sugar. Fourth, the outer edges of the nitrogen-containing bases are available for potential hydrogen bonding. Here's a picture showing the structure of DNA. The chemical structure of RNA. Each RNA strand is composed of nitrogenous bases covalently bound to a sugar phosphate backbone. It is a single-stranded molecule, unlike DNA. The sugar in RNA is ribose instead of deoxyribose, and the four nitrogenous bases are adenine, cytosine, uracil, and guanine. There are a variety of forms of RNA. Messenger RNA is a copy of a section of DNA that serves as a template for the manufacture of one or more proteins. Transfer RNA binds to both mRNA and amino acids and brings the correct amino acid into the growing polypeptide chain during protein formation, based on the nucleotide sequence of the mRNA. Ribosomal RNA and proteins are the cellular components of ribosomes, the site in which proteins are built.
DNA replication and causes of mutation. Although DNA replicates with fairly high fidelity, mistakes do happen. Fortunately, most mistakes are fixed through various DNA repair processes. But some replication errors make it past these mechanisms, thus becoming permanent mutations. These altered nucleotide sequences can then be passed down from one cellular generation to the next. If they occur in cells that give rise to gametes, they can be transmitted to subsequent organismal generations. Moreover, when the genes for the DNA repair enzymes themselves become mutated, mistakes begin accumulating at a much higher rate. In eukaryotes, such mutations can lead to cancer. Fixing mistakes in DNA replication. DNA polymerase ensures that the bases added to a growing strand are correctly paired with the complements on the template strand. However, DNA polymerase makes mistakes at a rate of 1 in every 100,000 nucleotides. Fixing mistakes. Proofreading is the ability of DNA polymerase to remove and replace incorrectly paired nucleotides in the course of replication. Mismatch repair processes that corrects mismatched nucleotides in DNA after replication has been completed. Enzymes excise incorrectly paired nucleotides from the newly synthesized strand and use the original nucleotide strand as a template when replacing them. Other means of DNA alterations. Environmental damage and spontaneous changes. Spontaneous refers to the fact that changes occur in the absence of chemical radiation or other environmental damage. Rather, they are usually caused by normal chemical reactions that go on in cells such as hydrolysis. As with replication errors, most environmentally induced DNA damage is repaired. The same is true of spontaneous mutations. Errors are a natural part of DNA replication. Tautomeric shifts. Biologists initially speculated that most replication errors were caused by what are called tautomeric shifts. Both the purine and pyrimidine bases in DNA exist in different chemical forms or tautomers, in which the protons occupy different positions in the molecule. Scientists believe that if and when a nucleotide base shifted into its rare tautomeric form, a likely result would be a base pair mismatching. Wobble. Most DNA replication errors are caused by mispairings, either between different but non-tautomeric chemical forms of bases, like bases with an extra proton which can still bind but often with a mismatched nucleotide, such as A with G instead of a T, or between normal bases that bond because of a slight shift in the position of the nucleotides in space. This type of mispairing is known as wobble. It occurs because the DNA double helix is flexible and able to accommodate slightly misshaped pairings. Slippage. Replication errors can also involve insertions or deletions of nucleotide bases that occur during a process called strand slippage. Newly synthesized strands may loop out a bit, resulting in the addition of an extra nucleotide base, or the template strand loops out a bit, resulting in the omission or deletion of a nucleotide base in the newly synthesized or primer strand. Wobble-induced mismatched base pairs. Strand slippage, insertion, and deletion. The scenario on the left shows addition, and the scenario on the right shows deletion. DNA replication postulates. There are the semi-conservative, conservative, and dispersive methods of DNA replication. However, only the semi-conservative has been found biologically significant. Semi-conservative DNA replication model. According to the semi-conservative model, after one round of replication, each new DNA double helix would be a hybrid that consisted of one strand of old DNA bound to one strand of newly synthesized DNA. Then, during the second round of replication, the hybrids would separate and each strand would pair with the newly synthesized strand. Afterward, only half the new DNA double helices would be hybrids. The other half would be completely new. Every subsequent round of replication, therefore, would result in fewer hybrids and more completely new double helices. Straight or circular? To date, no one has found any evidence for either conservative or dispersive DNA replication. Scientists have found, however, that semi-conservative replication can occur in different ways. For example, it may proceed in either a circular or a linear fashion, depending on chromosome shape. Bacteria with circular chromosomes undergo theta replication because the structure generated resembles the Greek letter theta. This process results from the original double-stranded DNA unwinding at a singular spot on the chromosome known as the replication origin. As a double helix unwinds, it creates a loop known as a replication bubble, with each newly separated single strand serving as a template for DNA synthesis. Replication occurs as the double helix unwinds. Eukaryotes undergo linear replication, as their cells carry vastly more DNA than bacteria do. Eukaryotic chromosomes have multiple replication origins, with multiple bubbles forming.
DNA transcription. The instructions a cell requires in order to sustain itself is referred to as the genetic code. The instructions stored within DNA are read in two steps, which are transcription and translation, which takes place in the nucleus and cytoplasm respectively. Transcription begins at the coding regions, which are bounded by promoters, which are regions on the DNA that show where RNA polymerase must bind to begin transcription of RNA. Here is a diagram of the coding regions of a template strand bounded by promoter region and termination site. In prokaryotes, transcription initiation takes place at a region called the Tata box. Once transcription is initiated, the DNA double helix unwinds and RNA polymerase reads the template strand, adding nucleotides to the 3' end of the growing chain. Also, the complementary strand now contains the nitrogenous base uracil instead of thymine. Terminator sequences are found close to the ends of coding sequences. In eukaryotes, termination of transcription occurs by different processes, according to the type of RNA polymerase used. mRNA processing. After DNA is transcribed into RNA, editing must be done to the nucleotide chain to make it functional. Introns, which are non-coding regions of DNA, are spliced out of the sequence by a spliceosome, which are large, complex macromolecule machines composed of protein and small nuclear RNAs. Exons, which are segments of DNA that code for the actual proteins, are rejoined by the enzyme DNA ligase. A guanine triphosphate cap is added to the 5' end of the newly copied mRNA, important for stability while undergoing translation. Polyadenylation takes place at the 3' end of the mRNA, consisting of about 250 adenine nucleotides, important for prolonging the lifespan of the strand. In this diagram, the pre-mRNA strand is being edited as the intron is spliced out by the spliceosome and the exons are joined together by the enzyme DNA ligase. Translation. During translation, the mRNA is read according to the genetic code, which relates the DNA sequence to the amino acid sequence in proteins. The translation machinery, called the ribosome, is located in the cytoplasm. The ribosome is composed of two subunits, the large subunit and the small subunit, which form a complex onto the mRNA strand where translation occurs. The small subunit of the ribosome has 3 binding sites: an amino acyl tRNA site, a polypeptidyl site, and an exit site. The P and A sites are important for allowing attachment of 2 tRNA molecules. tRNA molecules are special adapter molecules which possess one end that can read the triplet code in the mRNA through the complementary base pairing. Enzymes called amino acyl tRNA synthetases attach the correct amino acid to each tRNA based on the 3D structure of the tRNA molecule. Here is a diagram of the tRNA molecule containing anticodons to match the codons of the mRNA strand for a particular amino acid. Initiation. 3 initiation factor proteins known as IF1, IF2, and IF3 bind to the small subunits of the ribosome. They play important roles, one of which is keeping the large ribosomal subunit from prematurely binding. A methionine carrying tRNA then bind to the mRNA near a AUG start codon, forming the initiation complex. The large ribosomal subunit then joins the newly formed complex. Here is a diagram showing the important formation of the ribosomal complex, the site of translation in cytoplasm. Elongation. As ribosome moves along the mRNA in the 5' to 3' direction, 2 tRNA molecules with their amino acids move into the A and P sites of the ribosome. Polypeptide bonds are subsequently formed through peptidyl transferase activity, joining the amino acids together. These peptide bonds continually form until one of the 3 stop codons is reached.
The central dogma of molecular biology suggests that DNA maintains the information to encode all of our proteins and that there are 3 different types of RNA that passively convert this code into polypeptides. Specifically, messenger RNA carries the protein blueprints from a cell's DNA to its ribosomes, which are the machines that drive protein synthesis. Transfer RNA then carries the appropriate amino acids to the ribosomes for inclusion in the new protein. Meanwhile, the ribosomes themselves consist largely of ribosomal RNA molecules. Functions of RNA. Although RNA has been primarily associated with protein synthesis, there has been found to be catalytic RNA that carry out biochemical reactions similar to enzymes, and there have also been found to have complex regulatory roles. Non-coding RNA. RNA and tRNA are integral in translation. Small regulatory RNA, sRNAs. Small nuclear RNA. These molecules play a critical role in gene regulation by way of RNA splicing. snRAs are found in the nucleus and are typically tightly bound to proteins in complexes called snRPs. Small nuclear ribonucleoproteins, sometimes pronounced SNRPs. MicroRNA, miRNAs, have been shown to inhibit gene expression by repressing translation. Small interfering RNA also work in inhibiting translation. Small nucleolar RNA are molecules which function to process rRNA molecules. The significance of non-coding RNA. The complexity and diversity of RNA forms and activities in both prokaryotes and eukaryotes lends credence to the so-called RNA world hypothesis. This hypothesis states that RNA may have evolved prior to DNA and protein, and it may have played the roles of both of these molecules in the earliest life forms. The fact that some RNA have both coding and catalytic capacity without the need for a protein-based enzyme makes such a hypothesis viable. Nevertheless, it remains unclear whether today's RNA molecules with catalytic proteins are remnants of the evolutionary past or whether they have more recent origins. The ongoing discovery of new small regulatory RNA molecules suggests that additional functions may yet be uncovered. Thus, the full contribution of RNA to the life of the cell may still be unknown.